welcome to the Director's Desk, the podcast where we spotlight the groundbreaking work of the National Institutes of Health and the extraordinary individuals advancing innovation in biomedical research and public health. I'm Dr. Bhattacharya, Director of the NIH, and today I'm honored to be joined by Dr. Jeffery Taubenberger, Acting Director of the National Institute of Allergy and Infectious Disease, also known as NIAID. Jeff, thank you for coming on the podcast. Really glad to have you.
Thanks so much. It's an honor.
Okay, so we should start with the elephant in the room. You are sitting as the acting director of NIAID, a position that was once held by Tony Fauci.
Yes, for about 40 years.
So you were there as a researcher for 20 years, not focused on coronaviruses but on flu viruses, right?
Right. I mean, my lab's main interest for about 30 years has been influenza, and but during COVID, like most other people, we
that
turned to
happened to
COVID.
me too, yes.
And we became kind of a full-time coronavirus lab for a while. But while we still have coronavirus interests, we're still mainly a flu lab, and we've done other things. We've done some Ebola work, and mostly high containment virology is what we do.
Right. And what was that like for the first, let's say, up until the pandemic?
Yeah, well, that was really fantastic. I mean, I was recruited to set up a new lab division in the intramural program to focus on pandemic flu and influenza pathogenesis using state-of-the-art high containment facilities here in a new building on the NIH campus, and it was just really fantastic. I was first in the Cancer Institute for about 6 years, and then I was at Walter Reed at the Armed Forces Institute of Pathology for 13 years, and then I have been here now for 20 years. And so it's just been a great career. I believe strongly in public service and wanted to give back. And this was an opportunity to do really unique things using high containment virology that we had great labs and great support and great facilities. So it's just been a fabulous place.
So you were free to, you had this interesting project on the 1918 flu. You're working now on a universal flu vaccine with a technology that's, in some sense old, but also in some sense quite innovative.
Yes. Yeah. So, our work on influenza, I didn't start out thinking that we were going to make a vaccine. I started working on influenza was kind of an accident. It was a fluke. I picked it up as a hobby project when I was at the Armed Forces Institute of Pathology. At that point, I was leading a department of what was called molecular genetic pathology, starting in the 90s, when basically that area of clinical pathology was being invented, in which we could, instead of just looking at something under the microscope to make a diagnosis of cancer or an infectious disease, you could actually do genetic tests at PCR, Southern blot, and other things. And so we applied that to the kind of way that tissues are always processed standardly, still now, where you have formalin fixed, paraffin embedded, where the sections are cut for pathologists to look at them under the microscope.
Okay, well, hold on. Yep. This is a family-friendly podcast. Let's break that down. Paraffin embedded. So paraffin is like candle wax. Okay.
So what happens in pathology is if you want to look at a piece of tissue under the microscope, you have to slice it very thinly, about the thickness of a red blood cell. So very, very thin. And you have to get it on a glass slide, and you have to stain it with dyes to pick up different colors so that then you can look at it under the microscope. So this process has been invented, since the mid-19th century, and it's been basically was completely refined by the late 19th century. And they use something that they call fixatives. They started with alcohols, and now they use aldehydes, formaldehyde or other things, and that it just preserves the tissue. It kind of stiffens it. And so you take a little piece of tissue and you actually embed it in molten wax, and then you put it on a little kind of bologna slicer, and you can slice these little sections, and you put those on glass slides, and you stain them with what you want. So that's,
And so you can see what happens if there's like an infection, for instance, how the cells respond to infection.
Exactly.
And what, right?
So, there are times where, you can make a diagnosis, say, of a certain kind of tumor, a sarcoma or a lymphoma, but you might not be able to understand all the molecular details as we get more toward personalized medicine. So we can find that there are very specific oncogene mutations or very specific translocations that actually would dictate the kind of therapy that you would want to get. So it was really useful and very cool, cutting edge stuff. And I guess it still is cutting edge as advanced. So, I became a molecular genetic pathologist. But so in the mid-90s, we were sitting at an institution that was started by Abraham Lincoln during the Civil War. It had the largest collection of pathology in the world, samples going back to the Civil War. And I was trying to think, we have these new fantastic tools that allow us to look at things in a new way that have never been done before. What could we do to show how cool this technology is? And so the idea that I had, the crazy idea, was that we could find tissues of people who died of the Spanish flu in 1918 that had been preserved for, at that point, about 85 years or so, and see if we could use these new molecular techniques to fish out tiny fragments of the virus's genetic material.
But they're formalin fixed in this paraffin thing, so there's basically the viruses are dead.
Oh, the viruses are completely dead.
Yeah.
They're blown up into a million pieces.
Okay.
Most of the
so there was not any risk of any lab leak or anything like that.
Oh no. This was there was no infectious material at all, and
right.
The virus is blown up into teeny tiny the RNA into teeny tiny fragments.
But it is really cool to be able to see what was the biological entity that caused such devastation in 1918.
So the reason we picked that is that I remember just one line in med school. The Spanish flu was really bad, but that's about it. And I didn't know that much about it. But as I started reading about it, you become really hooked because it was really devastating. The scholarship on how many people died around the world keeps going up because the prior accounts starting from the 1920s usually just included the first world. And didn't really include the developing world. And now as we add more, I think a reasonable number of people who died in less than a year's time is about 50 million, maybe 100 million, out of a population of about 1.8 billion people on the planet. So there are some estimates that as many as 17 million people died on the Indian subcontinent. Really horrible. More than 40% of U.S. soldiers that died in World War I of all causes, more than 40% died of Spanish flu. These are healthy 18 to 25-year-old men, most of them in training camps here still in the U.S. We were just really gearing up to go to the Western Front. And so it was devastating. The funny thing was that, or the sad thing, is that because of the advances of microbiology by 1918, with the germ theory, the advances in even some vaccinations, that they thought this was going to be the first war where infectious diseases would not be the major killer. And
of course it was.
Nature proved us wrong again.
So now, fast forward to today, actually, you're working on a universal flu vaccine.
Right. So this comes from my strong interest in the Spanish flu. Where did the virus come from and why was it so bad? And that led ultimately to sequencing the whole genome using methods then that took us a decade in the lab that I can now replicate in my lab now in about a week and a half. So 10 years of effort into a week and a half thanks to technology. But then we rebuilt the virus to actually make an infection clone, and that's where you get into the infectious part, where we have to do that as a select agent with a lot of oversight.
Select agent means what?
It's a pathogen identified either as a very significant risk pathogen for humans or for animals or plants. And so the USDA has a list of agents for animals and plants, and the CDC has a list of agents for humans, and then there's some overlap agents.
Ebola, for instance, would be
Ebola
for humans.
Yes, exactly. Yeah. Typically, bird flu in the past, the 1918 flu, once it's reconstructed. And they can only be worked with under very great scrutiny and with a lot of permissions and oversight.
I visited one of the labs that the NIH runs. It's called a BSL-4 lab at the Rocky Mountain Lab in Montana. And when you work in one of these labs, you're working in a spacesuit, basically.
Right. So, there are different levels of biocontainment that the U.S. government helps design.
This was the highest level.
Right, this is the highest level. And so for influenza, because there are antivirals, because there are vaccines, it doesn't meet the BSL-4 level. So it's what we call BSL-3 enhanced. So we don't have to wear the spacesuit, but we wear a full sort of disposable outer Tyvek suit with kind of like a bicycle helmet with a shield, which has a positive air pressure.
I have a confession to make, Jeff. I'm a very clumsy lab person, which is why I went into epidemiology and health policy. But
obviously, everyone has to be very well trained, lots of supervision, lots of oversight at all levels. But the whole reason is that there are so many. When you see these labs, you see there's so many levels of engineering and thought goes into why it's safe because we have so many redundancies. I mean, any one thing would probably provide a lot of protection. What you're wearing, the gloves, double gloves, working inside a biosafety cabinet, the airflow, but you put everything together, plus the biosecurity, plus the clinical oversight of the people. Everything that goes into this is what makes it possible to work with this safely. Obviously, no one who works in these labs wants to infect themselves, or more importantly, infect their family or their friends or their grandmother, right? So great care is taken to do this kind of work safely. And I say that, people say that they don't like the idea of high consequence pathogen research, but the reason we use the term high consequence is that these are pathogens of high consequence. Ebola has a 50 to 60% case fatality rate. The 1918 flu killed tens of millions of people. H5N1 bird flu, are dangerous pathogens that could
but
cause
there are
pandemics.
let's try to do the less controversial thing. So let's talk about the 1977 flu epidemic, worldwide epidemic. There's pretty good evidence that was a lab leak.
Well, I don't know that it's a lab leak, but it was probably an iatrogenic thing. So let me explain.
Okay.
So in 1976, in Fort Dix, New Jersey, a couple of U.S. soldiers got very sick with influenza, and one of them died. And when they did analysis of the virus, based on what they could do in 1976, they realized that it was a swine influenza virus. And let me just go back in time, 1918, just for 1 second, is that the 1918 pandemic went into humans, probably from birds. That's our research has shown that it was an avian virus that adapted to humans sometime before the pandemic. Then humans gave the virus to pigs, and it subsequently adapted to pigs right in the fall of 1918. And so since 1918, you've had the 1918 virus evolving in two separate hosts at different paces. It evolved much more rapidly in humans, and it evolved more slowly in pigs, because pigs don't, in a sense, live long enough to develop this kind of long population immunity because of what happens to pigs. And so the pig virus stayed more 1918-like because it evolved slower than the human virus. There was less antigenic drift. So when they identified these infections with a pig virus in 1976, the expert opinion at the time was this is the reappearance of the 1918 flu. We already know how bad the 1918 flu was, so they said, my gosh, if we have another 1918 pandemic, it could be devastating. So there was an effort to make a vaccine very rapidly, as you know. President Ford was convinced to do that. They made this vaccine.
No lockdowns, though.
There were no lockdowns.
Okay.
A number of other countries thought, gee, maybe we should prepare. My understanding, and this is limited information, and I think we're all limited, is that in China there was some concern. And so what is a reasonable hypothesis, can't be proven, is that they may have done a vaccine challenge experiment. And that
who's they? First, they. Who's they?
In China.
The China. And the vaccine challenge. Explain that.
And so, where you would give, you would make a new vaccine, and you want to see how well it works. You could do it in the community in a traditional kind of phase 2 type study in a natural community setting, or you could do it experimentally, actually, like we do here at the NIH with Matt and Emily, where we actually take very carefully screened healthy volunteers and we intentionally infect them with wild-type modern flu viruses to be a model of self-limited illness for flu. It's a very efficient way of testing whether a vaccine or a new drug could be efficacious. You don't need thousands of people. You can do this, say, with 100 people.
But you only do it with agents that are, I mean, most of the time you get flu, you're not going to, you're going to recover from it.
So, in 1976, the virus that circulated really wasn't that pathogenic. Basically, because H1N1 viruses had been in humans now from 1918 up to 1976, basically everyone had immunity, except for people born between 1957 and 1977. There was a 20-year gap. And so only people under the age of 20 were really susceptible. Everyone else had immunity.
1957 was the last time there was an H1N1.
There was a pandemic in 57 that switched the subtypes of the virus, and then another one in 1968. And H1 then went away at 1957, but it came back in 1977. And now what we know is when you sequence the virus that was first isolated in 1977, it absolutely genetically matches viruses from the early 1950s. And there's no way we can think of biologically in a natural setting, even an animal or anywhere else, where an RNA highly mutable virus would stay the same. So the genetic evidence is super strong that it was a historical virus that had to have been grown up, that had been collected in the 50s, and that had been grown up. How it actually happened is not clear, but it does seem like it was a human-based activity.
It resembled the old version of the virus that had been collected fixed and so on, yeah.
Sat in a freezer. So, I mean,
Right.
just as an example, the 1957 pandemic is so-called H2N2, without getting into all the complications of flu naming. But
That's another podcast.
that's another puppy. The problem with that is that since 1968, nobody on Earth has seen an H2 virus. That virus was completely human adapted. It's not a select agent. We work with it under the same conditions as we would the 1918 virus, but that's not an obligation. And it probably sits in clinical diagnostic and basic lab virology freezers all over the world.
Yeah.
And it really scares me, actually, because I think we concerned about how a new flu pandemic could occur. But here's one that actually was a pandemic. Now, the vast majority of the world's population born after 1968 does not have immunity to H2. And so, that's something that I worry about.
Right. Okay, so let's go back to your career at the NIH. We talked about the time
Right.
up to the pandemic.
Right.
I want to talk a little bit about the time during the pandemic. And what was that like?
Well, it was a very difficult time for everybody, of course, all over the world. We had the shutdowns. We were, at the same time, trying to, do what we can to do really good work on the coronavirus pandemic. I put together a large multi-center autopsy study as a pathologist. I thought that one of the most important things I could do would be to help us understand how the virus is behaving in people, especially with serious illness and those who succumb to the illness and have fatal outcomes, and so have autopsies. And we did really amazing, sophisticated kind of systems biology study looking at all aspects of that, and we learned a lot of really interesting things, which we could talk about. But then we started thinking about developing new vaccine strategies. Based on, you had previously asked, but I forgot to answer, we had been working on ways to develop new generation flu vaccines that provide protection against not just one strain, but multiple strains. So not that you wouldn't need to have the vaccine applied every year. And in a pandemic, we don't know what the virus is going to be, so we can't predict it in advance, so we don't have a vaccine in advance. So the idea would, could you make a vaccine that would provide protection, at least from serious illness and death, in any case, whether it's seasonal flu or an infection from an animal virus like H5N1 or a real pandemic virus. So we had been working on that, and we applied that to our coronavirus question. And you see how quickly the coronavirus adapted and evolved, like it is sort of akin to flu, so that these new variants kept appearing, and the vaccines, weren't keeping up. And we're always, same with flu, we're always chasing behind that. We're not in front of it. So
it's really tough in the middle of a pandemic to develop a vaccine quickly and make sure that it's the correct strain is being addressed. And
it's very difficult, especially
when
we had limitations of how many people. Only about 20% of our lab could work at one time, and it was just very difficult to schedule. So it was very challenging to do all that. But obviously, we did some good work. But it was a difficult and kind of scary time.
Was it? Were you under a lot of obviously pressure to internal pressure to want to address the pandemic? But I guess what I'm trying to say is was it challenging from the outside? I tell you, it was challenging because as I said, I work mostly on epidemiology, health policy. But at one point, I decided I was going to early on in the pandemic do a seroprevalence study, a measure of how many people had antibodies to the flu, to the coronavirus. That was in April 2020, and there's a lot of I had a result that there was a lot more widespread than people realized very early on in the pandemic. Actually, I learned that our common friend Matt Memley, who's the acting director of the NIH, did a very similar seroprevalence study with very similar results. I found it difficult on the outside, even as a Stanford professor, to get those results out in front of the public without facing tremendous internal pressure to essentially to stop talking. Did you face any of that, or did you notice any of that?
Yeah, so there's something that is a little troubling about science, and it's not specific to this, but I'm going to make a more general comment, which is that scientists and physicians who do science should be always very skeptical and critical of data, right?
That's our job.
The science cannot be settled. Science is the pursuit of knowledge which never ends, right? And so all you can say, and the reason that I'm not good at reporters don't like it when they interview me.
I'm enjoying this. I'm not a reporter, though. So.
But a reporter wants a headline. And I always couch everything, well, this is what we think now, and this is what we don't know, and here's what we still need to do, because that's how I think all the time, instead of just giving a punchline. But what I was going to say is that scientists are susceptible to what I would call groupthink. And there are sort of scientific ideas that become very widespread and are accepted. It would be unkind to think what I just was thinking, but I'll say it's kind of magical thinking sometimes, that people accept things without necessarily looking at the data. I always try to remember to be a very careful scientist. And so, there are certain things that just sort of become part of the ether. They develop the way that people think about things. But when you look at the data, you find that the data are not necessarily true. So especially in a novel situation, there was a lot of that. But there's a lot of that in science just in general, and we have to always be very careful about that.
I mean, this is one of the reasons why I think that free speech and academic freedom are so important for for progress in science.
Right.
It's the only way we can combat groupthink.
Right.
Like we allow people to say, no, Jeff, I don't think that's right. Maybe you should check that one more time.
Yeah, absolutely. No, I agree with you completely. There's a quote, Max Planck, the famous Nobel Prize-winning physicist, has a quote from more than 100 years ago that said that a new idea in science is not accepted based on its merit. It's because the people who don't believe it eventually die. And another group of generation of scientists come up and they just learn this.
Science advances one obituary at a time.
Yeah, so
right?
it's sad that that science is not living up to what science should be. I mean, we should always be skeptical. We should always be open to new data. So, I mean, there was a lot of that because, people were trying to respond based on the advice that was being given, and it became a little difficult.
Okay, so let's turn to a more pleasant topic. Well, let's talk about pandemic preparedness. And you showed me before this podcast a book that you found from the 19th century.
Yes.
You care to share?
Sure.
In fact, I think it's sitting next to you.
It is sitting next to me. So I brought this for show and tell, if that's okay, in your podcast. This is a book I keep on my shelf because it's really
By the way, folks, Jeff is fluent in German.
So this is a book that was written in 1820.
He could read the script, you guys.
Yeah, it's a textbook. Yeah, I won't make you read it, but it's a textbook of influenza written by a doctor in Germany in 1820. And on the front page, he said the whole point of this book, and I'll quickly read, is to answer the following questions: what is influenza? Where did it come from? How did it behave in the past? In what ways can we predict how it will behave in the future? And how can we treat it? And how can we stop its spread? So those questions that this guy, Dr. Most, asked in 1820. Are the questions that I ask in my lab and many other people ask today, more than 200 years later. And it's very humbling because we have an enormous amount of science. We have sequenced millions of influenza genomes. There are 1 billion papers from very basic science to clinical science that have been published in influenza. There are labs all over the world who've studied this virus intensively for 80 plus years, 90 years now.
And we can't answer those questions.
And we cannot answer those questions. That doesn't mean those questions aren't worth trying to answer. But we are not at a point yet. We do not have the fund of knowledge to predict even what next year's seasonal flu is going to be like, much less when the next pandemic will occur. So in my view, the only thing we can do is react to a pandemic when it happens, sadly, but that's the truth. But what we can do, and what I've always focused, is that nature ultimately controls these things. And if we try to understand how pandemics in the past worked, maybe we could try to understand if there are some basic ground rules. So my interest in 1918 or past pandemics has been that. How did an avian virus become a human-adapted virus in 1918? What can we learn from that? What are the lessons from the 1957, 68, 2009 pandemics?
But nature is complicated, right? So it is going to be very difficult. It's not like, even weather forecasting is difficult.
Correct.
There's a version of pandemic preparedness, which I think became an industry over the last couple of decades, where you go out into the wild places, find, like, for instance, take in 2003, I think it was, the SARS-1 virus sort of emerged, seemingly out of nowhere. It turned out it was a animal origin, right? In China. There were outbreaks in Toronto. There were outbreaks, a few other places around the world, including the U.S. Luckily, it didn't spread very far because it was, for coronavirus, is quite deadly.
It was very deadly. We were very lucky that it didn't spread more. It easily, I think, could have caused a pandemic.
Yeah, and we don't really understand fully why it stopped.
Correct.
We don't. So people were very concerned for a long time that there might be another coronavirus that comes out of maybe China. There was another one in the Middle East called MERS, I think in 2008-ish. I forget exactly. So people were very concerned to be able to predict if there was going to be emergence of a new coronavirus related pandemic. And the sort of research program involved going and finding coronaviruses in bat caves and elsewhere, bringing them into labs, often not with the kind of track record of safety that you talked about. For instance, the Wuhan lab in China, and experimenting with them. What's your view of that kind of research program? Because pandemic preparedness of the sort you say, I completely agree with. Like, how do we, as a society, prepare for the fact that we don't understand exactly when these things are going to emerge? I mean, things like let's have our population be metabolically healthy, right? That's a good way to deal with the pandemic, because if metabolically healthy populations are resilient in face of new infections potentially.
Right. It's a difficult problem, but it is an important problem. Just like saying predicting. You mentioned the weather, predicting hurricanes or tsunamis or something, right? We can't predict them. We can react to them, but that doesn't mean we can't do some things that help us better prepare for the unexpected, right? That changing the code in which the houses and buildings are built, and so on, things like that. So we can certainly think of how to develop better response strategies, how to develop new therapies that might be broadly protective against even viruses we don't know yet, vaccines, therapeutics, other non-pharmaceutical.
Although we have to be careful with that, because these are for vaccines developed against viruses and pathogens we haven't seen yet. You can't test them on humans in advance.
Correct.
And the version that you have prepared for, because you brought it out of a bat cave or something, may not be the version that emerges.
Right. No, this goes back to what was in the book here, is that we absolutely cannot predict what will happen. I think that there is certainly something to say, like in defense of the coronavirus thing. We have 4 cold viruses that are coronaviruses, 2 alpha viruses, 2 beta coronaviruses that cause, sort of minor colds.
C43 and those other,
right. They all came from animals. They got into humans, adapted to humans, and probably caused pandemics. Turns out that there was a really bad pandemic in 1889 that's been called the Russian flu. It's always thought of as an influenza pandemic. But when you look at this beta coronavirus, this OC43, when you do the regression, it suggests that it entered humans around 1890. So there's just this at least hypothetical suggestion that a coronavirus may have caused a pandemic in 1889. That's not clear. I'm trying desperately to find fixed tissues from that time period to try to prove or disprove that theory, but we haven't found them yet. I think such tissues exist. Just the problem is nobody knows where they are. So there's real reason to suggest that coronaviruses can cause pandemics in the past, have caused them outbreaks. Obviously, we had SARS-1, which disappeared.
But OC43 now is a cold virus.
Correct. And SARS-CoV-2 is evolving in a way that it's much less virulent from the combination of population immunity and viral evolution. Again, viruses don't have intention. Obviously, they're not even technically alive, right? But it's much easier to think of them, at least from my limited brain power, as an anthropomorphic way. I like to sort of think about what they're trying to do. Now, admit that this is a fake setup, but what a virus like an influenza virus, it's beneficial if it wants to replicate itself in a host and then spread to another host.
It's almost impossible not to anthropomorphize
right. So
wants.
If it kills the host so quickly that it can't be spread to another host, there's a real strong negative selection pressure against that, right? So there are probably clear evolutionary pressures to reduce virulence and pathogenicity.
Although that's not guaranteed, but it's a tendency,
there was a
right?
tendency. One just quick aside: the interesting thing about the 1918 flu is that the changes that made the virus so pathogenic in humans are not a mutation specific to the pandemic, but something that was just accidentally inherited from a bird, where it's not pathogenic at all. And it's just like Ebola. Ebola isn't trying to be a human virus. It just, by accident, happens to be really pathogenic. So, I think that in SARS-CoV-2, that we're seeing less pathogenicity now. But in terms of your question, knowing where all the coronaviruses are in the world is obviously an interesting question. How big is this virus family? But being able to say, how can we manipulate them in a way that would predict their future behavior is something that is less likely to yield positive results because there are so many complexities that we don't understand.
Right,
And,
but yet that
and
was
it
that
also,
was that was the research program, right?
Right. And it was done in China and in a different kind of regulatory
BSL-2 lab, much less stringent than, for instance, the one you use here for flu viruses. Okay, so let's get to the last part of our conversation today. Well, hopefully you'll be back so we can talk some more fun stuff. But the kind of research that we've been talking about, that you've just been talking about, it is often been characterized as gain-of-function work, dangerous gain-of-function work. And there was a recent executive order by the president essentially ordering us as scientists to do a calculation. Before we do any research of a sort that might end up causing catastrophic harm to human populations, we should ask ourselves, should we do that? Like we should we should have independent folks who aren't us, the scientists themselves, often want to do the research, even if you can tell yourself lots of stories that if this is completely safe, I'm going to keep it completely safe. But we ought to have like the other independent eyes look at it and say, is this worth doing? Right. So for instance, move it out of biology into nuclear physics. So Enrico Fermi, when he launched the nuclear age by starting the first nuclear chain reaction on a squash court, I guess, in the University of Chicago. He did a calculation in his head, on paper, saying, what's the probability that this nuclear chain reaction will engulf the world? The probability was basically 0, and then he started the thing. The old way of regulating this kind of research, the kind of research you just talked about, where you take the virus and you essentially play with it to give it more functionality, was to say, are you on a list of select agents that you're working on? Are you working on something that's really dangerous? Are you working on Ebola? Then are you working to make Ebola more transmissible among humans or more dangerous? Then you subject yourself to this framework. And often the framework was not, actually the old framework that we had, it was called the P3CO framework, which I'm sure you know all about, is, I think through its whole history from 2017 to 2025, saw a total of 4 proposals. 3 proposals. Okay, 3.
I sat on the committee.
You sat, okay, well, there you go. So you saw 3. That doesn't seem like it's the right amount. It seems like there ought to have been more.
Yes, I agree. And I think that there was much about that oversight that needed to be improved. And, there's been lots of discussion in the community to improve this. And I think the executive order gives us a great opportunity to revisit this question. This is critically important, that we need really good risk-benefit analysis. This ratio is really important. Science doesn't exist in a vacuum.
We should do the calculation that Enrico Fermi did before he launched the nuclear age.
Yes, and so these are, and it's really good that we have this kind of discussion. It's really helpful, and I look forward to seeing how this progresses and have hopefully our institute at NIAID will be able to play a role in this.
So in the first batch of regulatory evaluation of this, so just before we do that, let's just talk about the philosophy. Rather than looking at a select set of select agents that we know are pathogenic, we say, okay, you do a calculation. Is this experiment, does it have some possibility of causing catastrophe? Whether you're working on a select agent that's currently dangerous or not, right? And then that calculation forms the basis of the risk-benefit analysis you're talking about.
Right. I completely agree. The idea of making it sort of pathogen agnostic makes a lot of sense.
Right.
Because you should really looking at the potential outcomes, thinking of functionality. That's really, I think it's a very good approach.
Right. And so now in this first batch, there were something like 40 projects that were identified as where you're just going to do the committee, the group that's going to be doing the regulation, are going to just do that calculation. It doesn't necessarily mean you're going to stop the project. It just means let's do the calculation before we put the green light. Is it worth the risk?
Right.
Is there a risk? Is it worth it first?
Yeah, and the community had a similar, gain-of-function pause about 12 years ago, where that's what the government asked.
Community means the infectious disease community.
Infectious disease community, but on the behest of the United States government.
Right.
We put a gain-of-function pause in around 2012 while these initial policies were formulated. And this has been an ongoing issue for 10 or 12 years, and it's an iterative process. The issue is that it's not a simple black and white issue, right? That there is gray in between, and very reasonable, very well-informed people could fall on opposite sides of a line wherever you draw the line, right? And knowing that that's the case, having a wide variety of people with different levels of expertise, not just virologic expertise, but the safety, the national security, all sorts of other questions, having them weigh in on this is really important. And the problem the P3CO setup is that while it had those kind of people, it didn't have the teeth, in a sense, to do things. And we were actually not given the option to look at the risk-benefit ratio, which I think was essential. Without that, there was really no way to really evaluate. So, and I think the key thing here is that work on nasty bugs that have the potential to kill people, for which we want to develop better therapeutics, diagnostics, prognostics, treatments, and preventatives, need to happen. That's important for global health. It's important for U.S. health. Even for viruses that don't exist or other pathogens that don't exist in the U.S., it's important for the U.S., for our public health, to make sure they don't come here, like Ebola, like Lassa fever. But thinking about the research that's done that would change behavior of pathogens has to be done very carefully. And I'm not going to say that some people would say none of this work should be done. And I think that this framework is not suggesting that. The framework suggests that work needs to be evaluated at this risk-benefit ratio widely, and that there needs to be oversight on not only when should it be done, and if the answer is yes, how it should be done, where it should be done, under what oversight, under what specific conditions and criteria, and what outcomes and what monitoring. That's key. And so we need to make sure that this is not a black and white issue, that there are some times where this kind of work is probably going to be really important. And the U.S. government is going to admit that it's really important, and it has to be done safely. I favor this kind of work being done where possible in U.S. government labs by U.S. government scientists, monitored by U.S. government safety officials and regulators, and all that stuff, with all the openness and transparency.
Yeah.
So exactly.
But in order for that to happen, I mean, what you're talking about is really a culture shift within the infectious disease research community, right? A shift where that kind of risk benefit is seen as like good for science.
Yes.
I mean, I've sensed some resistance to that. Maybe I'm wrong.
I think that people will come along with this when they think about it. We've talked about in our institute just having kind of boilerplate language on grants that go out, whatever the topic.
Well,
is that
let me
if
an
you do an
analogy.
experiment, let me just quickly say that if you're doing something that might lead to an unexpected finding, that might lead to something that's potentially dangerous gain of function that you weren't even expecting, please stop that, notify us right away, and then we'll put this through the analysis. And I think that would be good. What you want is the community itself. Most of this research, of course, is done by extramural funders that are not here, and so we want people to be self-reporting. We want people to take charge of that and think about themselves.
Let me make an analogy to clinical research, right? In order to do any kind of clinical research, there's a whole infrastructure set up to protect human subjects, right? And I've done research that involved human subjects before. Mm-hmm. I took that very seriously.
Yes.
Like the regulation essentially is there, but even more importantly, the researchers that are doing this kind of research want to protect human subjects. Mm-hmm. Like it's part of the ethos of the community of people who do clinical research to protect human subjects and not do something that would jeopardize the lives of research participants or harm research participants in any way. And that's our ethical obligation when we do that kind of research. Mm-hmm. And it's very, very important when that not just that there's a police function of some regulatory committee outside that says you can or can't do something. That needs to be there. But also that the community itself desires to want to do that take that into account when they do any research, right? So you want to act like Enrico Fermi.
Yeah.
Do the calculation before you launch the nuclear age.
So the education and dissemination of this information is important. So it has to go to the investigators. But just like clinical research, local universities or other institutions have their own biosafety committees. They need to have really good clear guidelines so that they can learn how to screen applications, and so the investigators can learn that. But the first part of this screening is going to happen at the level where the science is being proposed.
Right.
Right. And then we have to have that here at the NIH, and then certain things probably can progress to whatever ultimately ends up being the decision-making mechanism for this new dangerous gain-of-function analysis.
And so the endpoint will be a scientific sort of infrastructure that allows us to address the real risks of infectious disease without having to worry quite so much about the conduct of that research leading to a pandemic itself.
Mm-hmm. Right. Right.
Well, Jeff, that's been fantastic talking with you. I've been looking forward to this for a long time, and I'm glad to have you join me today.
Well, thanks you very much, Jay. It's been a pleasure meeting with you and talking to you. Thank you.
To our listeners, thank you for tuning in. Drop your comments in the chat as to what subjects we should cover next. Please be sure to hit that like and subscribe button on your preferred podcast platforms. This is Dr. Jay Bhattacharya, the NIH Director, and this has been the Director's Desk. Until next time, thank you.